We are back, and today we are looking at something that completely shifted the way I view the world right outside my window. We're doing a deep dive into the anatomy of flowering plants. And honestly, before we started prepping for this session, I always looked at a tree or a flower and just saw, I just saw green,
Just a surface level aesthetics.
Exactly. You see the leaves, the bark, but when you actually strip all of that away and look at the biological matrix underneath, it is staggering. So welcome to our study group. It is just you listening, me, and our resident mentor who is going to guide us through what I am now convinced is the most complex plumbing and architectural blueprint on the planet.
It really is an engineering marvel.
Yeah.
And I'm glad you're approaching it from that angle. Because if you go into studying plant anatomy, especially if you're gearing up for a major exam like the Neet, and you just view it as a dry list of Latin names to memorize, you're going to get bogged down.
Oh, completely. It's just flashcard soup at that point.
Right. More importantly, you're going to miss the absolute genius of how these organisms function. To truly get it, you have to start by asking a fundamental question. What is the core biological problem that a plant has to solve?
Well, the most obvious one to me is that they are completely stationary. Like, if a storm rolls in, I can run inside. If a drought hits, an animal can migrate to a river. A plant just has to sit there and take it.
Precisely. They are rooted to the spot. So imagine you are tasked with building a skyscraper. But this skyscraper is alive. It has to build itself from the ground up, starting as a single microscopic entity.
Right.
It has to grow hundreds of feet into the air to compete with other skyscrapers for sunlight. It has to pump thousands of gallons of water straight up against the unrelenting force of gravity. And has to synthesize its own food using just sunlight, water, and air.
All while withstanding, hurricane-force winds and freezing winters and scorching summers.
Exactly. How do you design an internal architecture that survives all of that without ever taking a single step?
When you frame it like that, it makes you realize that their survival isn't about running or hiding. It is entirely dependent on their internal structure, their plumbing, their structural steel, their waterproofing.
That is exactly what this topic is really about. We're looking at how a plant builds itself to survive. And because the material we are covering today is incredibly dense, we need a solid mental map for you listening. We aren't just going to throw vocabulary at you.
Please don't.
We won't. We're going to follow the logical flow of construction. First, we'll examine the individual bricks, the tissues. Then we'll look at how the plant organizes those bricks into an infrastructure, the tissue systems.
Okay, tissues, then systems.
Right. From there, we'll walk through the actual floor plans of the roots, the stems, and the leaves. And finally, we will look at the renovations, how a fragile green sapling undergoes secondary growth to transform into a massive wooden tree.
The bricks, the infrastructure, the floor plans, and the renovations. That makes so much sense. I want to start right at the foundation, the bricks, the actual tissues that make up the plant. When I was looking through the source material, the very first distinction it makes is dividing all plant tissues into two broad categories based on one simple ability.
And that ability is division. Can these cells multiply, or are they done multiplying? A tissue is simply a group of cells that share a common origin and usually perform a common function. But the plant needs two very different workforces.
It needs builders, and it needs the building itself.
Exactly. It needs a workforce to actively build new structures, and it needs a workforce to actually be the structure.
So the builders are the meristematic tissues, and the finished structures are the permanent tissues. I was thinking about meristematic tissues, and it's kind of wild. They are basically the perpetual youth of the plant. They are the stem cells that never grow up. They just keep dividing and dividing.
That's a great way to conceptualize it. In humans, once we reach a certain age, our fundamental growth in height stops. Our cells specialize. But plants have these localized, specialized regions of active cell division called meristems that allow them to grow essentially forever. And where these meristems are located dictates how the plant grows.
The text highlights three main types: apical, intercalary, and lateral. Apical seems pretty straightforward. They sit at the apex, right?
Yes, the extreme tips. You have the root apical meristem at the very bottom of the root, driving it deeper into the soil to find water. And you have the shoot apical meristem at the very top of the stem, pushing the plant higher into the air to capture sunlight.
So they just stretch the plant vertically.
They do. These apical meristems produce the primary tissues of the plant. They are responsible for the primary growth, which is an increase in length. Now, here's an interesting detail. As that shoot apical meristem surges upward, it leaves tiny clusters of cells behind.
Oh, like leaving a small construction crew behind on the lower floors while the main elevator keeps going up?
I love that analogy, yes. Those left behind cells form what we call an axillary bud. These buds sit right in the axil of a leaf. That's the angle between the leaf and the stem. And they essentially sit dormant until the plant decides it needs a new branch or a flower. They are biological insurance policies for future growth.
That makes perfect sense for the apical meristems. But the second type, the intercalary meristem, that one really caught my attention. The sources mention that these occur between mature tissues, and the classic example they use is grasses.
This is a phenomenal evolutionary adaptation. Think about a lawn or a vast savanna. Herbivores are constantly grazing, coming along and ripping the tops off the grass.
Right, and if the only place a plant can grow from is the apical meristem at the very tip, and a cow just chewed off that tip well, the grass should be permanently stunted. It should never be able to grow taller again.
But it does. And it does so because of intercalary meristems. Instead of relying solely on the tip, grasses have these zones of actively dividing cells located lower down on the stem, wedged right between the fully mature non-dividing tissues.
So when the top gets grazed off, or sliced by a lawnmower, the intercalary meristems kick into overdrive and regenerate the parts that were removed.
Precisely. It's like a hidden backup engine.
Go ahead and eat the roof. I can build a new one from the second floor.
It is brilliant survival architecture. Now, both apical and intercalary meristems are classified as primary meristems because they appear early in the life of a plant and are responsible for building the primary plant body, getting it tall and getting its initial leaves out. But what about the third type?
The lateral meristem. My understanding is that if primary meristems are for height, lateral meristems are for girth. They make the plant wider.
Spot on. Lateral meristems, also known as secondary meristems, appear later in the life of a plant. They mostly show up in plants that produce a woody axis, like trees and shrubs. Instead of being at the tips, they are cylindrical. They run up and down the length of the mature roots and shoots.
So they wrap around it like a sleeve.
Exactly. When they divide, they add layers outward, increasing the thickness of the plant. Examples include the fascicular vascular cambium, intercalary cambium, and Cork Cambium. But we will dive very deeply into those when we get to the renovation stage of our blueprint.
Okay, so we have our builders, the meristems. But eventually, a cell has to graduate. It divides, it's pushed away from the meristematic zone, and it has to take on a permanent job.
Right. Following cell division, the newly formed cells become structurally and functionally specialized. They undergo profound changes in their cell walls and their internal protoplasm. Once they reach maturity, they generally lose the ability to divide. They become what we call permanent tissues.
And we divide these permanent tissues into two distinct categories, simple and complex.
Which feels pretty intuitive.
Yeah.
Simple tissues are made of only one type of cell, and complex tissues are a mix of different cell types working together. Let's look at the simple tissues first. There are three main building materials the plant uses here: Parenchyma, Collenchyma, and Sclerenchyma.
I was trying to map these to human construction materials to make them stick in my head.
That's the best way to do it. Let's start with Parenchyma. This is the most abundant tissue in a plant. It forms the major component within all the organs. How did you visualize this one?
I kept thinking of it as the basic, fleshy packing peanuts of the plant, just the standard, versatile filler material.
That is highly accurate. Parenchyma cells are generally isodiametric. If you break that word down, iso meaning equal, diametric meaning diameter, means they are roughly the same width all the way around. They can be spherical, oval, polygonal, or elongated.
And their walls are very thin, right? Made of cellulose.
Yes, they have thin, flexible cell walls. They are living cells with a lot of cytoplasm. Sometimes they are packed tightly together, but often they have small intercellular spaces, little air pockets between them.
So what do they actually do?
Because they are this versatile living filler, they perform all the generalized functions of the plant. If they have chloroplasts, they do photosynthesis. They store food. They handle secretion. The soft flesh of an apple you eat, that's almost entirely parenchyma. The green, squishy part of a leaf, parenchyma.
Okay, so parenchyma is the living filler, the packing peanuts. But a skyscraper made of packing peanuts is going to collapse under its own weight. It needs support. This brings us to the second simple tissue, collenchyma.
Right. If parenchyma is the fleshy filler, think of collenchyma as flexible cartilage, or like rebar inside a concrete pillar. It provides structural support, but specifically, it provides flexible structural support.
I noticed the text mentioned it occurs in layers just below the epidermis in most dicotyledonous plants, but the structure of the cells is what's really unique. They aren't uniformly thick.
That is the key defining feature of Collenchyma. The cells are heavily thickened, but only at the corners. Imagine a box where the flat sides are thin, but the vertical edges where the sides meet are reinforced with thick layers of material.
Why just the corners, though? What's the biological advantage there?
It's an ingenious compromise between strength and flexibility. By thickening the corners, the cell gains significant tensile strength to resist pulling and bending forces. But by keeping the flat walls relatively thin, the tissue remains pliable.
What are they thickened with?
The thickening itself is due to a deposition of cellulose, hemicellulose, and importantly, pectin.
Wait, pectin, like the stuff used to make fruit jams and jellies thick and sticky.
A very same. Pectin is a complex carbohydrate that holds water and creates a gel-like flexible matrix. Collenchyma cells are living, they have no intercellular spaces between them, and their primary job is to provide mechanical support to the growing parts of the plant.
Like a young green stem or the petiole, the stalk of a leaf.
Exactly. Picture a young sunflower shoot in a strong spring wind. If its stem were entirely made of rigid, dry wood, the wind would just snap it in half. If it were entirely made of soft parenchyma, it would buckle and flop over.
So the collenchyma is what lets it whip around.
Yes. Collenchyma allows the stem to bend violently in the wind and then snap right back to a vertical position. It's the flexible cartilage that keeps the young plant intact while it's still growing.
That makes perfect sense for the growing phases. But eventually, a plant, especially a large one, needs actual unyielding structural steel to hold up massive branches and heavy foliage. And this is where we get to the third simple tissue, sclerenchyma. And I have to admit, when I was reading the source material on this, I had my first major whoa moment.
Sclerenchyma, the heavy-duty structural steel. What stood out to you?
The fact that these cells are dead, nature literally uses cell corpses to build the strongest structural pillars in the plant. Sclerenchyma cells build these incredibly thick, heavily lignified cell walls around themselves, and then at maturity, they usually completely lose their protoplast.
The living inside of the cell.
Exactly. They just suffocate themselves and die, leaving behind this massive hardened shell to act as internal scaffolding. It's almost a little macabre, but it is so incredibly efficient.
It is the ultimate biological efficiency. Think about it. Maintaining a living cell takes energy. The plant has to feed it, water it, deal with its waste. If the cell's only purpose is to be a rigid brick, why keep it alive once the brick is built?
That makes a lot of sense.
The key material here is lignin. Lignin is a highly complex, rigid organic polymer that makes the cell walls incredibly hard and compressive resistant. It's essentially the biological equivalent of pouring concrete.
And the sources break sclerenchyma down into two distinct shapes, fibers and sclerids.
Yes, depending on the form and structure, they serve different reinforcing purposes. Fibers are thick-walled, highly elongated, and pointed at the ends. They usually occur in groups or bundles.
Like stringy stuff.
Right. If you've ever tried to snap a mature stalk of celery and you get those tough, stringy threads that won't break, those are bundles of sclerenchyma fibers. They run lengthwise to provide longitudinal strength.
And then there are the sclerids. These sound completely different.
Sclerids are usually spherical, oval, or cylindrical. They are highly thickened dead cells, but instead of being long strungs, they are tight, hard little nuggets. Their cell walls are so incredibly thick that the internal cavity, the lumen, is reduced to almost nothing.
The examples the text gave for sclerids really grounded this for me. It mentioned the gritty texture you feel when you eat a pear or a guava. I always wondered what those little sand-like grains were. They're individual sclerids.
Exactly. They are microscopic stones embedded in the fleshy parenchyma of the fruit. Sclerids also form the incredibly hard shells of walnuts, the tough seed coats of legumes, and they are even found in the leaves of tea. Sclerenchyma is the plant's ultimate armor and weight-bearing framework, entirely constructed from the lignified skeletons of dead cells.
So, to summarize our simple tissues, our single-cell type building materials, we have parenchyma, the living, thin-walled, isodiametric packing peanuts used for basic filling and metabolism. We have collenchyma, the living, corner-thickened, pectin-rich, flexible cartilage for bending strength. And we have sclerenchyma, the dead, highly lignified structural steel used for rigid support. That is a fantastic toolkit for a self-building skyscraper.
It is. But a skyscraper is useless without plumbing. And that brings us to the complex tissues. These are tissues made of more than one type of cell working together as a coordinated unit. In plants, this is the vascular system, the xylem and the phloem.
The famous xylem and phloem. Xylem carries water up, phloem carries food down. Let's look at the xylem first. The text says it functions as a conducting tissue for water and minerals from the roots to the stem and leaves, but it also provides mechanical strength.
Because water transport requires rigid pipes that won't collapse under pressure. Xylem in angiosperms, the flowering plants, is composed of 4 distinct elements: tracheids, vessels, xylem fibers, and xylem parenchyma. Let's dissect the pipes themselves, the tracheids and the vessels.
Tracheids are described as elongated, tube-like cells with thick lignified walls and tapering ends. And just like Sclerenchyma, they are dead and without protoplasm.
Correct. Imagine a series of long, narrow, hollow needles stacked on top of and next to each other. Water moves from one tracheid to the next through tiny, unthickened areas in their walls called pits. It's a slightly tortuous path, zigzagging up the plant, but it works.
But vessels are different. They seem much more advanced.
Vessels are the biological equivalent of modern plumbing pipes. A vessel is a long, cylindrical tube made of many individual cells called vessel members. These vessel members are joined end to end. Like tracheids, they are heavily lignified and dead at maturity. But the crucial difference is that their end walls are perforated or completely dissolved away.
So instead of water zigzagging through side pits, it just shoots straight up a continuous open pipe.
Exactly. Vessels are much wider and much more efficient at moving massive volumes of water quickly. The other 2 components of xylem are xylem fibers, which are highly thickened sclerenchyma cells that just sit there and provide extra structural support around the pipes, and xylem parenchyma.
Which are the only living cells in the xylem, right?
Yes. They are thin-walled, living cells that store food materials like starch or fat, and they help facilitate the radial conduction of water, meaning moving water sideways out of the pipe to the surrounding tissues.
Okay, I have a massive star in my notes right here regarding vessels. The sources specifically call out a major exception when it comes to gymnosperms like pine trees.
Listen closely to this, especially if you're studying for the Neet exam. This is a classic high-yield concept that examiners love to test. The presence of vessels is a defining characteristic feature of angiosperms. Gymnosperms, the cone-bearing plants, absolutely lack vessels in their xylem.
Wait, so a massive, towering redwood tree is pulling all its water hundreds of feet into the air without the efficient, wide-open vessel pipe.
That's right. They rely almost entirely on the narrower and tapered tracheids for water transport. It's a more primitive but highly robust plumbing system. If an exam question asks, which of the following is absent in the xylem of gymnosperms, the answer is always vessels. Do not forget that exception.
Got it. Gymnosperms equal no vessels. Now, the text also introduces a concept regarding how primary xylem develops. It talks about protoxylem and metaxylem in these 2 terms. Endarch and exarch. I found visualizing this a bit tricky. Can we break that down?
Let's simplify it. When primary xylem is being formed by the apical meristem, it happens in 2 phases. The very first xylem elements to mature are called the protoxylem, proto meaning first or original. As the plant continues to grow, it forms later, usually larger xylem elements. These are the metaxylem, meaning after or later.
So protoxylem is the early stuff, metaxylem is the later stuff.
Correct. The critical detail is how these 2 are arranged physically in relation to each other. And that arrangement is entirely different depending on whether you were looking at a stem or a root.
Let's start with the stem.
In stems, the protoxylem, the early stuff, is formed towards the very center of the stem near the pith. As growth continues, the later metaxylem is formed further out, towards the periphery. This arrangement is called endarch. Think endo, meaning inside. The earliest, oldest xylem is on the inside.
Okay, so stem equals endarch, earliest on the inside. And roots?
In roots, the exact opposite happens. The protoxylem is formed towards the outer periphery, and the later metaxylem is formed towards the center. This arrangement is called exarch. Think exo, meaning outside. Earliest xylem on the outside.
Okay, we need a memory anchor for this because I can easily see myself flipping these 2 in a high-pressure situation. Here is the logic I used. Exarch equals exits outward. Roots reach outward. They're constantly trying to exit the center of the plant and push laterally out into the soil, so roots are exarch.
I love that. Exarch exits outward, roots reach outward. Therefore, roots are exarch, meaning protoxylem is on the outside. Write that down. It is a fantastic bridge to prior knowledge that will save you when the terminology gets confusing. So that's the water plumbing. Now let's look at the food plumbing. The phloem.
Phloem transports food materials, primarily sucrose, usually from the leaves where it's synthesized, down to the roots and other parts of the plant. And just like xylem, angiosperm phloem has 4 main components.
Yes. It is composed of sieve tube elements, companion cells, phloem parenchyma, and phloem fibers. Let's look at the main transport channels. The sieve tube elements. They're long, tube-like structures arranged longitudinally. Their end walls are perforated in a sieve-like manner, literally looking like a kitchen strainer, to form what we call sieve plates.
And here's where I found another incredibly strange biological fact. The text states that a mature sieve element possesses a peripheral cytoplasm and a large central vacuole, but it completely lacks a nucleus. It is a living cell doing complex transport work, but it has no brain. How does it survive?
It survives by outsourcing its brain work. This is one of the most elegant symbiotic relationships at the cellular level. Sieve tube elements are always physically and functionally associated with companion cells.
The companion cell is the brain.
Exactly. Companion cells are highly specialized parenchymatous cells. They are tightly connected to the sieve tubes by tiny channels called pit fields that run right through their adjoining longitudinal walls. Because the sieve tube threw away its nucleus to create more open space for sap to flow, the nucleus of the companion cell takes over.
That's crazy.
It controls the functions of the sieve tube. Specifically, the companion cells help maintain the pressure gradient within the sieve tubes, which is what actually pushes the heavy, sugary sap through the plant.
So the sieve tube is just a hollowed-out cargo pipe, and the companion cell is the dispatcher sitting right next to it pulling the levers.
That is fascinating.
Oh.
What about the other two components? Phloem Parenchyma are just elongated, cylindrical cells with dense cytoplasm and a nucleus. Their main job is to store food and other substances the plant makes, like resins, latex, and mucilage. Finally, we have phloem fibers, also known as bast fibers.
And based on the name fibers, I'm guessing these are sclerenchymatous.
You're starting to see the patterns. Yes, they are made of sclerenchyma. They're generally absent in the primary phloem, but are found in the secondary phloem. They are much elongated, unbranched, and have very thick cell walls. At maturity, these fibers lose their protoplasm and become dead structural support.
So they just turn to stone, basically.
Pretty much. In fact, phloem fibers are highly valuable commercially. When we harvest jute, flax, or hemp to make ropes, burlap, or linen clothing, we're actually harvesting the sclerenchymatous phloem fibers of those plants.
We wear plant plumbing support structures. Wild. Okay, but my Neet trap radar is going off again. We established a major gymnosperm exception for the xylem. Is there one for the phloem?
There absolutely is. Here is what you must remember. Gymnosperms do not have sieve tubes, and they do not have companion cells. Instead of sieve tubes, they have simpler structures called sieve cells. And instead of companion cells, they have albuminous cells.
No vessels in xylem, no sieve tubes or companion cells in phloem. They really run on older hardware.
They do. And examiners love to mix and match these exceptions to catch you off guard. But there's one more trap hidden in the phloem section. Phloem parenchyma, the storage cells, are absent in most monocotyledons.
Wait, so a grass or a lily, which are monocots, don't have phloem parenchyma?
Correct. A classic test question will ask, which of the following is absent in the phloem of monocots? And they'll sneak phloem parenchyma into the options. Or they'll ask you to identify the specific paired cells in pine trees. Know your exceptions inside and out.
Wow, okay. That is a lot, but it flows logically. Let me try to recap section 1, the brucks, before we move on. Plants grow using meristems, apical for length, lateral for girth, intercalary for grass backups. When cells graduate, they become simple tissues, parenchyma for fleshy filling, collenchyma for flexible bending support, sclerenchyma for dead, rigid steel support.
Spot on.
Or they become complex tissues, xylem for water, phloem for food, angiosperms use advanced vessels and sieve tubes, gymnosperms use primitive tracheids and sieve cells.
That is a phenomenal summary. You have the individual bricks mastered. Now, if you are a plant, you can't just have a pile of bricks. You have to organize them into functional zones. This brings us to section 2, the infrastructure. How do these individual tissues organize into larger tissue systems?
The sources state that based on their structure and location, plant tissues fall into 3 main systems: the epidermal tissue system, the ground tissue system, and the vascular tissue system. This feels like moving from cellular biology into macroarchitecture. Let's start with the outside layer, the epidermal tissue system.
Well, this is the skin of the plant. It forms the outermost covering of the whole primary plant body. It comprises epidermal cells, stomata, and epidermal appendages like hairs and trichomes.
The epidermis itself is usually just a single continuous layer of cells, right?
Wow.
Mostly parenchymatous.
Yes, tightly packed parenchyma with a small amount of cytoplasm lining the cell wall and a large central vacuole. Its primary job is protection. To help with this, the outside of the epidermis on parts exposed to the air is often covered with a waxy thick layer called the cuticle.
And the cuticle's job is to prevent water loss. But the text specifically notes that the cuticle is absent in roots, which, if you think about it for 2 seconds, makes total sense.
Explain why. If you are a root, what's your primary job?
To absorb water from the soil. If a root wrapped itself in a thick, waterproof, waxy cuticle, it would die of thirst. It would be like trying to drink water through a plastic bag.
Exactly. Structure dictates function. Roots have no cuticle. Now let's look at the most famous feature of the epidermis, the stomata. These are the microscopic mouths, mostly on leaves, that regulate transpiration, water evaporation, and gaseous exchange. Oxygen out, carbon dioxide in.
The stomata are pores, but they aren't just open holes. They are controlled by 2 highly specialized epidermal cells called guard cells.
And here's a massive, neat focus area, a classic textbook trap. You need to know the physical shape of those guard cells because it differs between plant types.
I have this circled in red. In dicotyledonous plants, like a bean plant or a sunflower, the guard cells are bean-shaped.
Yes, but in grasses, which are monocots, the guard cells are dumbbell-shaped. I guarantee you will see a question trying to confuse those two. Dicots equal bean-shaped. Grasses equal dumbbell-shaped.
How do they actually work to open and close the pore? The text mentions a difference in wall thickness.
This is pure mechanical physics. The inner wall of the guard cell, the side facing the stomatal pore, is highly thickened and somewhat rigid. But the outer wall, facing away from the pore, is thin and elastic. When the guard cells absorb water and become turgid, the thin outer walls bulge outward.
Like blowing up a balloon.
Yes. Because they bulge outward, they pull the thick inner walls apart into a crescent shape, opening the pore. When they lose water and become flaccid, the thick inner walls snap back together, closing the pore to save water.
It is an automatic hydraulic pressure valve. So the stomatal apparatus consists of the actual opening, the stomatal aperture, the 2 guard cells, and sometimes a few specialized surrounding epidermal cells called subsidiary cells.
Correct. Now what about the appendages, the epidermal hairs?
The text mentions 2 types based on location. On the roots, they are called root hairs. On the stems, they are called trichomes.
And you must know the difference between them. Root hairs are strictly unicellular elongations of the epidermal cells. They are just a single cell stretching out. Their job is to massively increase the surface area of the root to absorb more water and minerals from the soil.
But trichomes on the shoot system are usually multicellular.
Yes, trichomes are multicellular. They could be branched or unbranched, soft or stiff, and sometimes even secretory. But their main function is not absorption. They act as a physical barrier and a microclimate creator to help prevent water loss due to transpiration.
So root hairs absorb water, stem trichomes save water. Unicellular versus multicellular, keep those distinct. Okay, that's the epidermal system, the skin. Next is the ground tissue system. My notes refer to this as the meat of the plant.
It really is. The definition of the ground tissue system is simple. It is all the tissues of the plant, except the epidermis and the vascular bundles. It is the vast internal bulk, the filling between the skin and the pipes.
And because it's the filling, it consists mostly of simple tissues, right? Mostly our versatile packing peanuts, parenchyma.
Mostly, yes. You will find parenchymatous cells in the cortex, the pericycle, the pith, and the medullary rays of primary stems and roots. You might also find Collenchyma or Sclerenchyma in the ground tissue of stems for extra support. In leaves, the ground tissue is highly specialized. It consists of thin-walled chloroplast-containing cells, and is called the mesophyll. That's where all the photosynthesis happens.
Simple enough. Skin on the outside, pipes on the inside, ground tissue as the meat in between. So let's talk about how those pipes are organized. The vascular tissue system.
The vascular system consists of our complex tissues, the xylem and phloem. Together, they form discrete units called vascular bundles. And how these bundles are physically arranged within the plant is incredibly important. If I hand you a microscopic slice of an unknown plant part, the arrangement of the vascular bundles is the first thing you look at to identify what you are looking at. There are 2 main types of arrangement, radial and conjoint.
Okay, let's break those down. Let's look at radial first.
In a radial arrangement, the xylem and phloem within a bundle are not situated together. They are arranged in an alternate manner on different radii. Imagine a wheel with spokes. One spoke is a bundle of xylem, the next spoke is a bundle of phloem, the next is xylem, and so on. They alternate around the circle.
And where do we find this alternating radial pattern?
Remember our mnemonic from earlier. Exarch exits outward, roots reach outward. Well, here's another memory anchor for you. Root for radial. Radial vascular bundles are the defining characteristic of roots. If you see alternating radii of xylem and phloem, you are looking at a root.
Root for radial. That's perfect. So what is the other arrangement? Conjoint.
Conjoint means exactly what it sounds like, conjoined. The xylem and phloem are jointly situated along the same radius within the same vascular bundle. Instead of alternating spokes, they are stacked together. Usually, the phloem is located only on the outer side of the xylem.
And if radial is for roots, conjoint must be for the rest of the plant.
Correct. Conjoint bundles are the standard arrangement in stems and leaves. But within the conjoint category, the text makes another crucial division. Conjoint bundles can be either open or closed.
This relates to whether the plant can undergo secondary growth, right? Whether it can get thicker and form wood later on.
Exactly. In dicot stems, there is a very thin, specialized layer of meristematic tissue sitting right between the phloem on the outside and the xylem on the inside. That layer is called the cambium. Because the cambium is present, these bundles have the ability later in life to wake up and start forming secondary xylem and phloem. The potential for future growth is open. Therefore, they are open vascular bundles.
So dicot stems have open bundles, and monocot stems.
Monocot vascular bundles absolutely lack a cambium layer between the xylem and phloem. Because they have no cambium, they cannot form secondary tissues. Their growth in girth is physically closed off. Therefore, they are closed vascular bundles.
Open has cambium, closed has no cambium. Dicot equals open, monocot equals closed. This is all building so logically. We have our bricks, we've organized them into the skin, the meat, and the pipes. Now we are finally ready to look at the architectural floor plans. Section 3, roots, stems, and leaves. This is where we cut the plant open and look at a cross-section under a microscope to see how all these tissues are actually laid out in reality.
This is where we put everything we've learned together. Let's start from the bottom up, the roots. The hidden foundations. We need to compare the transverse section of a dicot root, like a sunflower, to a monocot root. Let's walk through the dicot root from the outside in.
Okay, starting on the very outside, we have the epidermis, which the text calls the epiblema here. Many of these cells protrude into unicellular root hairs. Then, moving inward, we hit a massive layer of ground tissue called the cortex, which is just several layers of thin-walled parenchyma cells with intercellular spaces.
Standard packing peanuts allowing water to flow through the root easily. But then, water hits the innermost layer of the cortex, the endodermis. And this is a structure you must understand deeply.
Yes, my Neet radar is absolutely screaming at me right now regarding the dicot endodermis. The text introduces a highly specific term here.
It should be screaming. This is a critical functional distinction. The dicot root endodermis comprises a single layer of barrel-shaped cells arranged tightly with no intercellular spaces. But here's the magic. Their tangential and radial walls, the walls facing the sides, not the inside or outside, have a deposition of a water-impermeable waxy material called suberin. This deposition forms distinct bands known as Casparian Strips.
Casparian strips made of waterproof suberin. Why is nature putting waterproof strips inside a root that is trying to absorb water?
Think of it as the ultimate security checkpoint. Roots are absorbing huge amounts of water from the soil. But dissolved in that water are all sorts of minerals, some useful, some potentially toxic. In the outer cortex, water just flows freely through the intercellular spaces between the parenchyma cells. It doesn't actually enter the cells. It just flows through the gaps. This is called the apoplastic pathway.
Like water flowing through the gaps in a pile of rocks.
Yes. But before that water can enter the vascular cylinder, the steel in the center, to be pumped up to the rest of the plant needs to filter it. It needs to control what goes up. The Casparian strips block those intercellular gaps. They act like waterproof caulk, sealing the spaces between the endodermal cells. When the water hits the endodermis, it can't go around the cells anymore. It is forced to actually pass through the living cell membrane of the endodermal cell.
Wow. It forces the water out of the free-flowing gaps and makes it pass through the cell's biological filter so the plant can selectively absorb what it needs and block toxins.
Exactly. It is a brilliant biological filtration system. Examiners will always test the keyword Casparian strips and the chemical suberin in relation to the dicot root endodermis.
Casparian strips equal suberin equal dicot root waterproof checkpoint. I will not forget that. Moving inward from the endodermis, we hit a layer called the pericycle.
Yes, a few layers of thick-walled parenchymatous cells. This layer is important because this is where lateral roots originate, and it's also involved in forming the vascular cambium during secondary growth later on.
And then we hit the very center, the vascular bundles. And we already know the rules here. From our mnemonic root for radial, we know the xylem and phloem are arranged alternately on different radii. And from exarch exits outward, we know the earliest protoxylem is on the outside periphery. But what is the actual difference between dicot and monocot roots at this level?
The primary difference is the numbers. A dicot root typically has very few xylem bundles, usually 2 to 4. This is called a diarch to tetrarch condition. Because there are only a few bundles, the very center of the root, the pith, is small or sometimes totally inconspicuous.
Okay, so dicots have 2 to 4 bundles, small pith. Now let's look at the monocot root.
Anatomically, the outer layers of a monocot root are identical to the dicot, epiblema, cortex, endodermis, pericycle. But look at the vascular bundles. Instead of 2 to 4, a monocot root usually has more than 6 xylem bundles arranged in a ring. This is called a polyarch condition. Poly meaning many.
And because there are so many bundles arranged in a wider ring, it leaves a lot of empty space in the middle.
Precisely. So the pith in a monocot root is large and well developed. And there's one more massive foundational difference you need to remember. We touched on it earlier. Monocot roots absolutely do not undergo any secondary growth. They never form a cambium ring. They stay exactly as they were built.
Diarch or tetrarch with small pith for dicots, polyarch with large pith for monocots, and monocots never get thicker. Okay, let's move up the skyscraper. The stems, the central highway. Let's start with the Dicot stem.
Okay, looking at a transverse section of a typical young dicot stem, the outermost layer is the epidermis, usually covered with a thin cuticle and bearing trichomes. Moving inward, we hit a massive zone called the cortex, which extends all the way to the pericycle. But in the dicot stem, the cortex isn't just one uniform layer. It has 3 distinct subzones. Let's focus on these, because they are full of exam keywords.
The outermost cortex layer sitting right beneath the epidermis is the hypodermis. And my notes highlight that in a dicot stem, the hypodermis consists of a few layers of Collenchyma cells.
Stop right there and think about why. We learned about Collenchyma in the bricks section. What does it do?
It's the flexible cartilage. It has thickened corners with pectin. It allows a young stem to bend in the wind without snapping.
Exactly. The dicot puts its flexible support right on the outside, in the hypodermis. Below that are regular thin-walled parenchymatous cortex cells with prominent intercellular spaces. And then we hit the innermost layer of the cortex, the endodermis.
Now, just like the root had a very special endodermis with the Casparian strips, the stem has a special feature too.
Yes. Here is what you must remember for the dicot stem. The cells of the endodermis are incredibly rich in starch grains. They act as a major energy reserve. Because of this, the endodermis in a dicot stem is often referred to as the starch sheath.
Starch sheath equals dicot stem endodermis. Got it. Moving further in, we hit the pericycle. Here, it is present on the inner side of the endodermis and above the phloem, but it's not a continuous ring. It appears as semilunar or half-moon-shaped patches of hard sclerenchyma. And then we reach the defining visual feature of a dicot stem.
The vascular bundles, how are they arranged?
A large number of vascular bundles are arranged in a neat, perfect ring around the central pith, like a pearl necklace.
The ring arrangement of vascular bundles is the characteristic feature of dicot stems. And based on the rules we established earlier, how would you describe these specific bundles?
Okay, let's use the logic. It's a stem, not a root, so it doesn't use radial. It uses conjoint, xylem and phloem on the same radius. It's a dicot, which means it can undergo secondary growth, so it must have cambium. That means the bundles are open. And since it's a stem, the earliest xylem is near the center pith, so it's endarch. So the full description is conjoint, open, and endarch.
Perfect deduction. That is exactly how you should think through it rather than just memorizing a string of words. Conjoint, open, endarch, arranged in a ring. Now let's completely flip the script and contrast that with a monocot stem. This is another area rife with neat traps.
Okay, looking at a monocot stem, like a piece of bamboo or a cornstalk, the very first sublayer, the hypodermis, is different right away. It's not flexible collenchyma, it is sclerenchymatous.
Yes. Nature uses hard, dead structural steel right under the skin rather than flexible cartilage. Monocot stems typically need rigid outer support quickly to grow tall fast without secondary thickening. What about the vascular bundles? Do they form a neat ring?
Not at all. They are completely scattered. There are a large number of vascular bundles scattered everywhere throughout the parenchymatous ground tissue. The bundles near the periphery are generally smaller and closer together, and the ones near the center are larger and more spread out.
A scattered arrangement is the hallmark of the monocot stem. Each of these individual scattered bundles is surrounded by its own sclerenchymatous bundle sheath. And because it's a monocot, what do we know about the bundles themselves?
They are conjoint, like all stems, but they are closed. Monocots don't undergo secondary growth, so they have no cambium.
Exactly. Closed bundles. Now, here are 2 very specific traps examiners will set for the monocot stem. I mentioned one earlier. Look closely at the phloem within these bundles. What is missing?
Ah, the exception we noted earlier. In monocots, the phloem parenchyma is absent. The food storage cells are missing from the phloem.
Yes. Trap number 1. Phloem parenchyma is absent. Trap number 2. Within the vascular bundles themselves, specifically in the xylem, there are large, distinct water-containing cavities. You do not see these in dicots. So if a question asks you to identify a stem with a sclerenchymatous hypodermis, scattered closed bundles, absent phloem parenchyma, and water-containing cavities, you are looking at a monocot.
That is a fantastic mental checklist. Dicot stem equals collenchyma hypodermis, starch sheath, ring of open bundles. Monocot stem equals sclerenchyma hypodermis, scattered closed bundles, no phloem parenchyma, water cavities. We have the foundation build, we have the central highway mapped. Now let's look at the roof. The solar panels, section 3, part 3, the leaves.
Leaves are incredibly fascinating because they are essentially massive flat sheets of complex tissue perfectly adapted to balance two directly competing needs, catching as much sunlight as possible for photosynthesis while preventing massive water loss from evaporation. Let's look at the dicot leaf first, which botanically is known as a dorsiventral leaf.
Dorsiventral. That means it has a distinct dorsal top and ventral bottom side right, like a typical oak or maple leaf. The top surface is usually a darker, waxier green, and the bottom surface is lighter and duller.
Exactly. The upper surface is called the adaxial epidermis, and the lower surface is the abaxial epidermis. Both are covered by a cuticle, but the placement of the stomata, the breathing pores, is highly strategic.
The text notes that the abaxial, the lower surface, generally bears significantly more stomata than the upper surface. The upper surface might even lack stomata entirely.
And why do you think that is? Imagine the physical environment of that leaf.
Well, the top of the leaf is baking in direct, hot sunlight all day long. If the plant put all its pores on the top surface, the heat would just suck the water right out of them. It would dry out instantly. By putting the stomata on the underside, they are in the shade, protected from the direct heat, which minimizes evaporative water loss.
It is brilliant structural shading. Now let's look between the upper and lower skin. This is the meat of the leaf, the ground tissue called the mesophyll, which is packed with chloroplasts and performs photosynthesis. In a dicot, or dorsiventral leaf, this mesophyll has two very distinct textures. It is differentiated.
Right. The upper part, sitting directly under the top epidermis, is called the palisade parenchyma. These are elongated, vertically arranged cells. They look like columns standing tightly packed next to each other, parallel to each other.
They are arranged that way to pack as many chloroplasts as physically possible into the zone that receives the most direct sunlight. They are the primary solar panels. But below them, taking up the lower half of the leaf, is the spongy parenchyma.
And these cells are oval or round and loosely arranged, with numerous large air spaces and cavities running between them.
Think about the stomata on the bottom of the leaf. They're opening up to let carbon dioxide in for photosynthesis. That air needs a place to go. The spongy parenchyma acts like a lung. The air enters through the stomata on the bottom, filters up through the massive cavities in the spongy layer, and reaches the tightly packed palisade cells above. Palisade on top for catching light, spongy on the bottom for distributing air.
Form perfectly following function. And the vascular bundles, the veins, are scattered through this mesophyll. In a dicot leaf, the veins form a reticulate or net-like venation.
Correct. The vascular bundles vary wildly in size depending on the size of the vein you are looking at. The main midrib has a massive bundle, while the tiny net-like offshoots have very small ones. Now let's compare that highly differentiated structure to the monocot leaf, also known as an isobilateral leaf.
Isobilateral means equal on both sides. Think of a blade of grass or a corn leaf standing vertically. It doesn't have a distinct flat top and bottom. Both sides are exposed to roughly equal amounts of sunlight as the sun moves across the sky.
Because the environmental pressure is equal on both sides, the anatomy adapts. What happens to the stomata?
Since there isn't a shady side, the stomata are present in roughly equal numbers on both surfaces of the epidermis.
And what about the internal meat, the mesophyll? Does it need a distinct palisade layer on top and a spongy layer on the bottom?
No, the text says the mesophyll is not differentiated into palisade and spongy parenchyma. It's just a uniform layer of identical photosynthetic cells all the way through the leaf.
Exactly. Undifferentiated mesophyll. Furthermore, because monocots generally have parallel venation rather than net-like, if you cut a cross-section, all the vascular bundles, except the main central vein, are nearly the same size.
Okay, so monocot leaf equals equal stomata, uniform mesophyll, same size veins. But here is where I had my second mind-blowing realization of the reading. It involves a specific adaptation in grass leaves called bulliform cells. When I read how these work, I was stunned. It is pure mechanical genius built into a cellular level. Walk us through the Bulliform cells.
This is truly one of nature's best hacks for surviving severe water stress. In grasses, certain epidermal cells on the upper adaxial surface, specifically along the veins, modify themselves into something new. They become quite large, they empty themselves of mostly everything, and they become colorless. These are the Bulliform cells.
They act like microscopic hydraulic hinges.
That's exactly how they function. Think of a long hinge running down the length of the grass blade. When there's plenty of rain, these large Bulliform cells absorb water and become fully turgid. They swell up and become stiff like a pressurized balloon. Because they swell up, they force the hinge open. This forces the grass leaf to unfurl, flatten out, and expose its maximum surface area to the sun for maximum photosynthesis.
But what happens during a severe drought when the plant is desperately losing water?
When there is water stress, the Bulliform cells lose their internal water pressure and become flaccid. They deflate. And because they are located along the top surface, when they deflate, the hinge collapses. This causes the entire leaf to physically curl inward on itself. It rolls up tightly like a tube.
By rolling up into a tube, the leaf hides its surface area, and more importantly, it hides the stomata on the inside of the tube, trapping the humid air and preventing any further water loss. It is an automatic hydraulic self-defense mechanism against drought.
It is brilliant. It requires zero conscious thought from the plant. It's just pure physics reacting to water availability. Yeah. If you ever walk past a field of grass or a cornfield on a scorching, dry summer day, and you notice that all the blades are rolled up tight into thin little needles, you know the soil is bone dry. The Bulliform cells have deflated to save the plant's life. It is a critical survival adaptation specifically for monocot leaves.
That is just so incredibly cool. Okay, so we have built the entire primary plant. We have our cellular bricks, we have our tissue systems, we've mapped out the roots, the stems, and the leaves, but our skyscraper is not finished. A young green sunflower sapling is one thing, but a massive towering oak tree with a trunk 10 feet thick is something entirely different. The primary growth got it tall, but how does it get so incredibly massive? This brings us to section 4, the renovations, secondary growth.
This is where plant biology gets truly dynamic. We established earlier that primary growth, driven by the apical meristems at the tips, is about increasing length. Secondary growth is exclusively about increasing girth, getting thicker. It's the process of turning a green shoot into a woody trunk, and this occurs primarily in dicotyledonous stems and roots, and in gymnosperms.
But not monocots. I feel like we need to hammer this home.
Let me repeat it clearly, because it is a foundational, non-negotiable rule in botany. Monocots do not undergo true secondary growth. They simply do not possess the cambium necessary to do it. They grow to their primary width and stop. Secondary growth is a dicot and gymnosperm phenomenon.
So how does this renovation project actually work in a dicot stem?
It requires the activation of two completely new lateral meristems, the Vascular Cambium and the Cork Cambium. Let's start with the inner renovator, the Vascular Cambium, which is responsible for building the wood. Let's think back to our dicot stem floor plan. We had our conjoint open vascular bundles arranged in a neat ring. And we said they were open because they had a thin patch of cambium sandwiched between the primary xylem on the inside and the primary phloem on the outside.
Right. That specific patch is called the intrafascicular cambium. Fascicular basically means bundle, so it's the cambium inside the bundle.
Exactly. But a series of disconnected patches isn't enough to add a solid layer of wood to the whole stem. The plant needs a continuous, unbroken ring of cambium, so it has to build the bridges between the patches. The cells sitting between the vascular bundles are the medullary rays. They are just regular, permanent parenchyma cells. But when secondary growth triggers, these specific cells do something amazing. They wake up, they become meristematic again, they regain the ability to divide.
They ungraduate and go back to being stem cells.
Yes, they dedifferentiate. This newly formed dividing tissue between the bundles is called the interfascicular cambium, intermeaning between.
So the intrafascicular patches connect with the newly formed interfascicular bridges, and suddenly you have a complete, continuous, active cambial ring running all the way around the stem.
The inner woodmaker is fully assembled. Once this continuous ring forms, it becomes highly active. It begins rapidly cutting off new cells, dividing both inwards toward the center and outwards toward the bark. The cells it cuts toward the inside, toward the pith, mature into secondary xylem. This is what we colloquially call wood. The cells it cuts toward the outside, toward the periphery, mature into secondary phloem.
But the cambium isn't an equal opportunity builder, is it? When I was reading this part, my notes literally say, this is a crushing reality.
It truly is a crushing reality. For reasons of biological necessity, trees need a lot of strong wood to hold themselves up. The cambium is generally much more active on the inner side than the outer. It produces a massive amount of secondary xylem, wood, compared to a relatively small amount of secondary phloem.
So think about the physics of this. You have a solid ring inside a stem, and it is rapidly producing dense hardwood on its inside. The center of the tree can't compress, so the expanding mass of wood pushes the cambium ring violently outward.
And as it pushes outward, what gets caught in the crossfire? Everything outside the cambium ring. The plant literally crushes its own primary phloem and older secondary phloem against the outer edge of the stem as the wood expands. They are gradually crushed to death by the plant's own growth.
That is brutal. It builds so much internal structure that it destroys its own outer plumbing.
It does. The primary xylem usually remains somewhat intact right in the very center, but the outer layers are obliterated. To keep at least some radial communication open between the inner wood and the outer layers, the vascular cambium occasionally forms narrow bands of Parenchyma cells that pass horizontally through the secondary xylem and secondary phloem. These are called the secondary medullary rays.
Okay, so the massive inner accumulation of secondary xylem is what we call wood. But if you've ever looked at a tree stump, you know wood isn't just one uniform color, it has rings.
Ah, the annual rings. This is where botany meets meteorology. The activity of the vascular cambium is not constant. It is under the strict control of physiological and environmental factors. In temperate regions, the climatic conditions change wildly through the year.
In the spring, the weather is usually wet and relatively mild.
Exactly. In the spring, the plant knows it's time to grow massive new leaves and branches. To support that, it needs to pump an enormous amount of water. So the cambium becomes highly active and produces a large number of xylem vessels. And specifically, it produces vessels with very wide cavities to maximize flow rate. This wood formed during the spring is called spring wood or early wood. Because the vessels are so wide and thin-walled, the wood overall is lighter in color and has a lower density.
But spring doesn't last forever. Eventually, autumn and winter arrive. Right. It gets cold, the ground might freeze, water becomes scarce. The plant needs to conserve energy and protect its plumbing from freezing and bursting. The cambium slows down dramatically. It produces fewer xylem elements, and the vessels it does make are very narrow and thick-walled. This is called autumn wood or late wood. Because the vessels are narrow and dense, this wood appears darker in color and has a much higher density. So in a single year, the tree produces a light, wide band of spring wood, followed by a dark, narrow band of autumn wood. Together, one light band plus one dark band equals 1 year of growth. One annual ring.
Precisely. Think of it like reading a tree's diary of the weather. Wide, light bands mean a good wet spring. A very thin, irregular ring might indicate a severe drought year, and by simply counting the alternating light and dark rings, you can accurately estimate the age of the tree. This science is called dendrochronology.
It's literally recording history in its own skeleton. But as a tree gets really old, say several decades or centuries, the wood right in the center changes again. The text mentions heartwood and sapwood.
Yes. The older a tree gets, the greater the mass of secondary xylem it accumulates. The oldest wood is sitting right in the center of the trunk. Eventually, the plant stops using this central wood for water transport entirely. Instead, it starts using it as a physiological dumping ground.
It dumps toxic waste into its own inner pipes.
It's a highly strategic toxic waste dump. The plant deposits organic compounds like tannins, resins, oils, gums, and aromatic substances into the central vessels and tracheites. These substances are dark, which makes the central wood appear dark brown. More importantly, these chemicals are highly toxic to microorganisms and insects. They make the inner wood incredibly hard, durable, and resistant to decay.
This dark, dead, toxic inner core is called the heartwood. It no longer conducts water. Its only job is pure, immovable mechanical support. It is the steel core of the mature skyscraper.
Exactly. When loggers cut down an old oak tree to make beautiful, dark, durable furniture, they are after the heartwood. But if the heartwood isn't conducting water anymore, how does the tree survive?
It relies on the newer wood on the outside.
Yes. The lighter peripheral region of the secondary xylem, the rings formed in the most recent years, is called the sapwood. The sapwood does not contain the dark tannins and resins. It is still biologically active, conducting water and minerals from the roots up to the canopy. So, heartwood is for ancient, rigid support. Sapwood is for active transport.
Okay, so the inside of the stem is expanding massively, piling on rings of heartwood and sapwood, pushing violently outward. But let's think about the outside of the stem. The epidermis, the skin, is only a single layer of cells. If the inside gets fatter and fatter, the outside is eventually going to split open like a tight shirt on a bodybuilder.
And that is exactly what happens. The outer cortical and epidermal layers get stretched to their breaking point and are physically ruptured by the expanding wood inside. If the plant leaves those tissues broken, it will dry out and die of infection. It desperately needs to replace its outer armor. Enter our second lateral meristem, the Cork Cambium, or phloem.
The outer renovator. Where does this one come from?
Usually, as the stem continues to increase in girth, a new meristematic tissue develops in the cortex region. This is the phloem, or Cork Cambium. It is a simple structure, just a couple of layers thick, made of narrow, thin-walled, nearly rectangular cells. And just like the vascular cambium deep inside, it starts cutting off new cells on both its inner and outer sides.
The cells it cuts to the outside differentiate into cork, or phloem. The cells it cuts to the inside differentiate into the secondary cortex, or phyloderm. Let's talk about the cork first, because humans use cork for a very specific reason. We plug wine bottles with it.
We do, and we use it because of its incredible biological properties. Cork cells are dead at maturity. But more importantly, their cell walls become heavily impregnated with a substance we've seen before, suberin.
Suberin, that's the exact same waterproof wax used in the Casparian Strips in the root endodermis?
Exactly. Nature recycles its best ideas. The plant pumps suberin into the outer cork cells, making them highly impervious to water and air. The cork forms a tough, thick, waterproof, protective outer shield. The inner layer, the secondary cortex or phyloderm, is just living parenchymatous cells. Together, these three layers, the phloem, the phloem, and the phyloderm, are collectively known as the periderm.
Phloem, phloem, phyloderm equals the periderm. Okay, now we need to address the ultimate definition trap in botany, the word bark. In everyday language, when I look at a tree, I call the rough, cracked brown stuff on the very outside bark. But botanically, bark means something very specific, and I know it trips up students constantly.
Pay very close attention to this because it is a favorite trick question on exams. Bark is a non-technical term, but in anatomy it has a very strict, inclusive definition. Bark refers to all tissues exterior to the vascular cambium.
All tissues. Let's trace that from the outside air going inwards to the wood. You have the periderm, the cork cambium, and the secondary cortex. Then, moving deeper, you have whatever is left of the primary cortex. Then what's next? The crushed primary phloem and the active secondary phloem.
Yes, that is the trap. Bark includes the periderm and the secondary phloem. Students constantly look at the dry outer cork and think that is the bark. They forget that the active secondary phloem, the living tissue transporting the plant's food down from the leaves, is physically located outside the vascular cambium, which means it is technically part of the bark.
So if you take a knife and strip a ring of bark completely around the trunk of a tree,
you aren't just removing its dead outer armor. You are completely severing its active secondary phloem. You are cutting the food supply line. The roots will receive no sugar from the leaves, and the tree will slowly starve to death. This process is called girdling, and it's a fatal wound to a tree.
ARC equals periderm plus secondary phloem. That is a vital equation to understand. Now, one more question about the bark. If the outer cork layer is completely impregnated with suberin, making it totally waterproof and airtight, how do the living tissues underneath in the stem breathe? Don't they need oxygen for respiration?
They absolutely do. The cork cambium doesn't create a perfect seal. It leaves little ventilation shafts. At certain regions, usually right under an old stomata, the phloem cuts off closely arranged parenchymatous cells on the outside, instead of the waterproof cork cells. These parenchymatous cells multiply rapidly and eventually rupture the outer epidermis, forming lens-shaped openings in the bark. These openings are called lenticels.
Lenticels. Lens-shaped breathing holes in the armor.
Yes. Lenticels permit the exchange of gases between the outer atmosphere and the internal living tissues of the stem. They are essential for keeping the massive trunk alive. So that covers the complex crushing process of secondary growth in the Dicot stem. Does the dicot root undergo secondary growth too? It does. It has to. Otherwise, a massive tree would be supported by thread-thin roots and it would topple over. A mature dicot root forms a woody structure very similar to the stem, but the origin story of its vascular cambium is completely different, and it is a major Neet trap.
Walk us through the trap.
Think back to the Dicot stem. The vascular cambium ring was formed from two parts. The intrafistula cambium, which was already there, making it primary, and the interfascicular cambium, which formed later, making it secondary. So the stem's cambium ring is partially primary, partially secondary in origin. But in the dicot root, there is no intravascular cambium. The vascular cambium is completely 100% secondary in origin.
Completely secondary. It builds it entirely from scratch later in life. Where does it come from?
It originates in the tissues located just below the primary phloem bundles and a portion of the pericycle tissue located just above the protoxylem. Because it connects these points, it starts out not as a circle, but as a wavy, continuous ring that weaves in and out between the alternating radial xylem and phloem bundles. As it starts dividing and adding wood to the inside, it eventually pushes outward to become a perfect circle. Once it's circular, the events are basically the same as in the stem-cutting secondary xylem inward to make wood and secondary phloem outward. But the origin is what matters for the exam. Remember, dicot root vascular cambium is completely secondary in origin.
A completely secondary wavy ring that eventually rounds out. Oh, wow. We have covered an immense amount of ground today. We started with microscopic, perpetually dividing stem cells, explored how they turn into packing peanuts and structural steel, watched them form complex waterproof checkpoints in roots, and finally saw them activate massive lateral renovations to build towering, woody, toxic, hearted tree trunks. It is a stunning biological narrative. But before we finish, we need to lock this in for the listener. Mentor, give us the things to remember, the ultimate memory anchors from this deep dive.
Absolutely. The biology is beautiful, but you need the high-yield facts ready to fire. Here's what you must remember. Number 1, the gymnosperm exceptions. Angiosperms have advanced plumbing, vessels in their xylem and sieve tubes with companion cells in their phloem. Gymnosperms lack all of these. They rely on primitive tracheids, sieve cells, and albuminous cells instead.
Number 2, the guard cells. Dicot stomata have bean-shaped guard cells. Classes, the monocots, have dumbbell-shaped guard cells.
Number 3, the layout rules. Use the mnemonics. Root-for radial roots have alternating radial vascular bundles. Exarch exits outward roots have exarch xylem, where the protoxylem is on the outside. Stems, conversely, have conjoint bundles and endarch xylem.
Number 4, leaf adaptations. Dicot, or dorsiventral leaves, have differentiated mesophyll-type palisade on top for light, loose spongy below for air. Monocot, or isobilateral leaves, have undifferentiated uniform mesophyll, but they possess bulliform cells, the massive hydraulic hinges that roll the leaf up to save water during a drought.
And number 5, the bark equation. Bark is everything outside the vascular cambium. Bark equals periderm, PLUS, secondary phloem. Never forget that the living food-transporting phloem is part of the bark.
Those are absolute gold. All right, let's do the 60-second sticky summary, the rapid-fire version of the entire plant architecture. Okay, plants build themselves from meristematic tissues, which actively divide, and permanent tissues, which are mature and specialized. Simple permanent tissues are Parenchyma for basic filling, Collenchyma with pectin corners for flexible support, and dead, heavily lignified Sclerenchyma for rigid steel support. Complex permanent tissues are xylem for upward water plumbing and phloem for downward food plumbing.
These tissues organize into 3 systems: epidermal, the outer skin, featuring stomata for gas exchange, and trichomes for water saving; ground, the meaty filling of cortex and pith; and vascular, the bundled pipes. Roots use radial alternating pipes. Stems use conjoint stacked pipes.
Looking at the specific floor plans, dicot roots have a suberin-laced endodermis forming Casparian strips, acting as waterproof biological checkpoints. Dicot stems arrange their open vascular bundles in a neat ring, protected by a Collenchyma hypodermis and a starch sheath. Monocot stems scatter their closed bundles randomly, protect them with hard Sclerenchyma, and lack Phloem Parenchyma entirely. Dicot leaves differentiate their internal meat to balance sun and air. Monocot leaves use hydraulic bulliform cells to curl up when they are thirsty.
And finally, the renovations. To get thick, dicots and gymnosperms activate vascular cambium to create massive amounts of inner wood, brutally crushing their own outer phloem in the process. They activate Cork Cambium to build outer waterproof periderm bark. Changing seasonal weather creates annual rings in the wood, and as the tree ages, the oldest inner wood is filled with toxins to become a hard, dead support core called heartwood.
Nailed it. What an unbelievable journey through the hidden architecture of a plant. You will never look at a tree or a blade of grass the same way again. But before we leave you today, I want to leave you with a provocative thought, a biological puzzle to mull over based on the strict rules we just learned. We hammered home the fact multiple times that monocotyledonous plants absolutely do not undergo true secondary growth. They lack the vascular cambium, their vascular bundles are closed. They cannot physically produce secondary xylem, which is what we call wood. So
Okay.
think about a palm tree.
Palm trees are monocots.
Exactly. Yet, if you look at a mature palm tree, they have massive, tall, incredibly thick trunks that look an awful lot like solid wood. If they don't have secondary growth, how on earth do their trunks get so incredibly thick and tall without forming true secondary wood? Take a close look at a palm tree next time you are outside, or pull up a picture on your phone, and think about how nature might have hacked its own rigid system to build a skyscraper without the standard renovations.
That is a fantastic puzzle to end on. Remember, knowledge is most valuable when you question it, stretch it, and apply it to the anomalies in the real world. Keep connecting the dots.
Thanks for diving deep into the blueprint with us today. Keep studying, keep observing, and we will catch you next time.